All right. Well, I'm going to go ahead and get started. Again, I want to thank the folks at St. Baldrick's Foundation for giving me the opportunity to talk to you this afternoon. My name is David Higgins. I work with the Children's Brain Tumor Network, and I'm here today to talk a little bit about the data resources and biospecimen resources we have to support researchers who are studying pediatric cancer, especially pediatric brain tumors. And so, as hopefully, or perhaps people in this audience probably already know, within pediatric illnesses, brain tumors are the leading cause of disease-related death in children in the United States, as well as numerous other countries around the world. And brain tumors are especially very difficult to treat because they're different patients than adult patients. Their brains are still actively developing and growing. And so treating children with treatments that have been developed for adults can result in very substantial and permanent side effects for these patients. Brain tumors are, of course, very complicated. So even if we have a drug in hand to be able to treat them, that drug might not be usable for multiple different tumor types. There are many different types of tumors. And in order to be able to develop these research discoveries and these targets, large institutions need to collect a large amount of samples to be able to carry out these investigations. And so it would possibly take years to be able to amass enough data to be able to lead to even a clinical trial where we're looking at a drug that could be used to treat a pediatric brain tumor. So this is the world that we're trying to work with, and this is the world that CBTN is really trying to fix and solve. So like I said, my name is David and I work at the Children's Hospital of Philadelphia. And today I'm speaking on behalf of the Children's Brain Tumor Network. So if you've never heard of us before, what we are, we're a network of researchers, clinicians, patients, and foundations from all over the world. And we're dedicated to creating a world where no child suffers or dies from a brain tumor. And so today, like I said, I'm so happy to be here with folks from the St. Baldrick's Foundation. Today, what I'm going to talk about is kind of have three main sections here. At the beginning, I'm going to go into some details about what CBTN is, an overview of who we are and where we came from and what we offer. And then in section two, I'm going to really be focusing on what types of resources are we sharing with investigators. And so we have a large number of different types of resources, and then I'll be able to go into a demonstration at the end to show how we use some of those resources to make discoveries. And finally, I'll wrap up with some next steps, places to go next, and ways that are collaborative, what we call our collaborative resources, so things to take with you at the end of the meeting today. All right, so who is the Children's Brain Tumor Network? So of course, patients are very critical partners to our success at CBTN. And so patients are ultimately making the decision to donate their tissue and samples to the larger community. So that way we can better understand and better learn more about brain tumor biology. And our goal, of course, is that from learning about brain tumor biology, we'll be able to develop cures faster. So CBTN has an IRB-approved protocol, and we enroll children and adolescents with any brain tumor diagnosis, ranging from most prevalent to very, very rare tumor types. And then we also follow each of these patients throughout their entire journey. So we want to track and collect information about the growth and evolution of their brain tumor so that we can build a valuable data set for researchers where we can ultimately lead us towards cures for these patients. And so leading back to my lead-in slides, brain tumors are, of course, very, very complicated. So there are many types of brain tumors, and each of those brain tumors have different subtypes. So within no two tumors are alike. And so, of course, tumors of different types have very different biologies. But then even within a single tumor inside a particular patient, the genetics of the cells within that same tumor might not be the same within a single collection. And so a drug that might target part of the tumor might not effectively treat the rest of the tumor. So tumors are very, very complicated in terms of how do we learn about them and how do we treat them in our patients. But of course, what's more, to complicate things further is that tumor tissue is a very precious and yet depletable resource. So the only way to really understand what's happening inside the tumor is to look at the tumor tissue more closely in terms of the genetics, in terms of the markers and things that are happening inside of the cells in the brain tumor. And so this finite depletable resource is very, of course, very significant that the patients are making this decision to contribute, but then also very, very limiting. And so the Children's Brain Tumor Network exists to help facilitate and collaborate and share with researchers around the world to maximize the contribution of these patients, sharing tissue and sharing data in order to be able to learn the most and get us to the most significant number of samples for analysis, which can lead to true breakthroughs and discoveries. And so we do that by connecting, harnessing, and empowering researchers around the world. And so we're very interested in a collaborative open science. So we realize that no single institution by themselves is going to make the breakthrough discovery. We really think that only through collaboration and sharing are these discoveries going to happen. And so by empowering investigators to be able to connect with each other, harness these data sets, and share through open science, can we accelerate research, accelerate clinical trials, and then ultimately accelerate clinical impact for our patients. And so, as I mentioned, the Children's Brain Tumor Network is a network of member institutions around the world. And so we started back in 2001 with just 4 sites based in the United States. And since then, in the last 11 years, we have had a huge number of growth. Now we have over 32 sites in 2022. And so we're working across the network with folks within these different sites who are willing to share biospecimens and share data across the sites. But even if you are not affiliated with any of these institutions here, you can still access CBTN data. So we're looking to connect with people no matter where you are. We want to share what's available. And our operations center, as I mentioned at the top, so I'm based out of our operations center in the Children's Hospital of Philadelphia. And so through collaborating, we've been able to have a larger impact than we could have had by working by ourselves here at CHOP. So we've been able to enroll almost 5,000 patients and make available more than 65,000 biospecimens. Working in our laboratory, our team of benchtop scientists have generated more than 150 preclinical models. And then through analyzing these tumor samples and DNA samples collected from patient parents, we've been able to genomically characterize more than 6,000 patients. All of this is resulting in new discoveries. And so we've made we've supported more than 307 different scientific projects, which have resulted in more than 89 new publications in the field. And so I don't, put these numbers up to necessarily brag, but I want to emphasize that, we support a lot of research and we're looking to continue that. We're not looking to rest on where we are. We want to continue to find new collaborators, new scientific projects, and support new discoveries in this field. Always looking to continue to share and collaborate with anyone who's willing to work with us. So CBTN starts with the patients, but we're also very generously supported by philanthropy. And so this process of collecting, storing samples, sharing samples, shipping them has been generously funded from 11 years of trust with member foundations who support our mission and support our work. And so this has allowed us to be able to share these resources available to any researcher around the world, as well as create what we call the Pediatric Brain Tumor Atlas, which is this the largest collection of clinically annotated pediatric brain tumor samples in the world. And so in order to be able to make this data available on the global scale, we have a partnership with the Gabrielle Miller Kids First Data Resource Center. And so this is an NIH-funded effort investigating pediatric cancer and structural birth defects. And so by partnering with the Kids First DRC, CBTN has been able to address this problem of how do we share this data? How do we make it available to more researchers around the world? And so if you're curious to learn more about the Kids First Data Resource Center, be sure to tune into the webinar tomorrow, where we'll talk a little bit more about Kids First and give some more details about that. And so again, we've been empowering scientific projects. And so we empower investigators, scientists, researchers, clinicians to be able to request and share our resources, which we categorize broadly into our specimens and our data. As we mentioned, we have thousands of specimens that we've provided to researchers across 20 different biospecimen projects to date. All of this is at no cost to investigators. So we're providing all of this to people who share our drive and share our mission of discovering breakthroughs for sick kids. And so again, to my final intro slide here is that we really believe that this sharing and open science model will help us accelerate breakthroughs. And so here we have kind of a comparison of a more traditional academic research model versus our CBTN collaborative model there on the right. And so, some steps are obviously going to maintain, the same amount of time in terms of testing and identifying, but we really think we can accelerate research at these steps here, this collecting sample data and developing or accessing cell lines. So what would normally take 6 to 7 years in terms of consenting patients at a single institution, in order to be able to have enough patients to be able to drive an investigative research study, CBTN has already enrolled patients. And so we have, across this network of hospitals, we already have patients who are willing to engage and willing to, help develop and discover a cure. And so it's years of time by sharing this network of patients who've already agreed to through consent to make these contributions. Additionally, in our laboratory, we're already working to develop, as many renewable cell lines as we can in order to be able to share these readily with investigators. And so this is already, being processed in our lab. And if these cell lines exist, we can deliver them within months instead of taking years to develop individually. So by collaborating, we really believe that we can help accelerate breakthroughs, make discoveries faster, and ultimately help children quicker as well. All right, so that's a little bit about who we are and our ethos and where we come from. And so hopefully it's clear that CBTN is, we are looking to work with investigators, share what we have, and help kids. So what do we have? And so this kind of leads me into my second topic here, CBTN's resources for researchers. And so one thing that we mentioned earlier was CBTN has biospecimens. And so CBTN's biospecimens, these are samples that are collected from patients or from their parents. And so we have many different types of biospecimens available. So for example, we have tumor tissue, which is available in a couple different methods of preservation. So we have flash frozen tumor tissue, but we also have formalin fixed paraffin embedded PEP, FFPE tissue as well. We also have isolated DNA and RNA that's available upon request. We have blood samples, as well as germline samples, typically saliva from patients. We also consent and collect samples from parents if they're willing to contribute in order to be able to look at heritability or track de novo mutations within a particular family. So we collect data from them as well as cerebrospinal fluid. Within or beyond the specific specimens that we collect from patients, as I mentioned, the specimens are finite, they're limited. And so there's only so many different projects that can be supported from a finite sample of tissue. So that's why we're very interested in developing preclinical models as well. So these would theoretically be able to be proliferated through time. And so we can, not just have the limited amount, but we can continue to grow cell lines that have this same tumor biology that can be maintained in the laboratory extensively or indefinitely. So we also make preclinical models available. We make cell lines, PDX, and organoids as well. And these are some numbers about the number of participants that we have biospecimens for and available to share. Attached to these biospecimens, though, we also collect clinical data. And so clinical data is information about the patients that we pair with these biospecimens. So of course, we collect the most basics, things like demographic, age, sex, race, and ethnicity about the particular patients. But beyond that, we also look to collect longitudinal data with family history. So we really want to learn, is there a history of these particular diseases, and again, track inheritance or possible inheritance as a means for where what might have caused the tumor to develop. We have information about the location of the tumor, and we continue to stay in touch and with the patients and check in on overall survival and progression-free survival to track patient outcomes. So additionally, we also make genomic data sets available as well. So CBTN provides omics level data. And so this is where we really find the strength of sharing. And so a lot of investigators, when we first started, were interested, I'm interested in receiving these samples so that way I can sequence the genome and then I can answer my question, et cetera, et cetera. And so we realized many people were asking those questions. And so this is where we began, well, why don't we sequence the genome and then we can share that with everybody. And so we can, the wonderful thing about the genomics files is that we can copy and paste them indefinitely. As long as there's storage space somewhere in the world, we can continue to share them with investigators and researchers. And so we do this by collecting a couple different types of this omics level data. So of course we do whole genome sequencing, and we make a couple different variant types available. So we do somatic variant calling or tumor normal calling. So this is where we'll compare the alignment of the brain tumor to the patient's alignment of a collection of normal cells, either blood or saliva, and then we'll be identifying what specific variants are unique to that tumor, what caused these particular cells to undergo this tumorous growth pathway. And so those are called somatic variants, so we make those available. We also call germline variants, so these are variants that are present in every cell of the patient's body. These are things that might be inherited or might predispose a patient towards developing cancer. We also, as I mentioned, we also collect family trios. And so if mom and dad agree and consent to join our protocol, then we will sequence their DNA as well. And then you can compare a trio of mom, dad, and patient to be able to track inheritance and track things like that. So we have lots of different whole genome data. We also have RNA sequencing data as well for looking at gene expression and gene fusion calls. We have a smaller subset of whole exome data, which provides similar information as whole genome sequencing in terms of the what's happening inside the DNA, as well as a subset of samples with proteomic data as well. And again, this is available across multiple brain tumor histologies. So a Children's Brain Tumor Network is not unique to a specific histology. We support any child with a pediatric brain tumor. And so here's a graph showing some of the more number of cases by tumor type shown there to the right. And so we're very excited. As part of CBTN, we actually received support from the National Institutes of Health, NIH, to do additional sequencing. And so we have been sequencing this past year an additional 5,700 additional samples that we're very excited to be sharing very, very soon, hopefully in spring 2023. So our data set is about to more than double in size. And once it is complete, we are going to share it with the world as soon as possible. So now is a great time to get involved and learn more about CBTN as we're ready to provide even more data for researchers. So how do we share this, and where does all this come from? And so in order to be able to access this, we share on what we call our genomic platforms. So we have 3 main sites that we use to share our genomic data. The Kids First portal allows investigators to be able to build cohorts of participants, identify these genomic files, these alignments, these variants, and then connect them over to Cavatica, which is our cloud-based research platform. So Cavatica is a powerful tool for being able to run bioinformatic pipelines, call your own variants, run analyses in interactive notebook environments with RStudio or Python, and then share these discoveries with researchers around the world. But we also support our third tool, which I'm a big fan of, is our tool called PEDC Bioportal. So in addition to making the raw genomes available and the raw VCF variant files available for investigators to be able to do their own genomic analysis on, we've begun the process of processing this data already, and we make it available on our tool PEDC Bioportal. So it allows investigators to be able to search genomic variants within specific CBTN samples. You can compare patient outcomes and other clinical data, and then investigate this intersection of genetic data and patient data as well. And so during our demonstration, I'm going to actually show PEDC Bio a little bit further. But here's a quick preview of what it looks like. And so when you log into the PEDC Bio portal, you can find the Pediatric Brain Tumor Atlas study, where it gives you information about this set of patients, tells you frequently mutated genes, and then you can continue to narrow it down by specific tumor type, specific histology, things like that. You can search for specific mutations, and so here is P53. And so this is a set of, a diagram here showing the length of the protein, and then each of these markers shows a particular mutation. And so provides information and annotation about each of the variants, within our different samples and within our different samples collected from our participants. And it highlights information about the genetics of these different tumors. You can also compare, and so here is looking at within these different P53 mutations across different tumor types, and so you can do your own kind of compare, ask your question, how does cancer type compare to number of mutations, and you can see roughly by looking at those for the P53 mutation have higher or on the higher side of the box and whisker plot across all histologies. So you can do more than just kind of search. You can actually begin to discover and then even download figures, which could then be entered into a publication. So you can begin your research as well. And then finally, it can provide a patient specific view. And so you can look specifically within a particular patient or within a particular sample. It will outline all of the different mutations that we observed, complete with annotations. So I'll show a little bit more about PEDC Bio in the future, but it's a very powerful tool. It's very interactive, all within the browser, in order to be able to make discoveries possible. The last resource I'll talk about is our imaging data. This is housed in a website called Flywheel. Imaging data, and so we are learning more and more about tumors, and often the first way that a clinician will learn about a tumor is by looking at the imaging data, or an MRI image of the patient. And so attached with many of these samples, we also collect these MRI images as well, in order to be able to learn how can we combine and these imaging data sets with the genomic data sets. And so is there a way to combine things that we're observing within the MRI in terms of the location of the tumor, the size of the tumor, the shape of the tumor? Are there things that would correlate with that in the genomics? And then based on that, what can that better direct clinicians to prescribe treatment? Can that better direct surgeons during operations? And so kind of combining the imaging with the genomics, this field of radiogenomics is something that we're very excited to be supporting as well. So all of our imaging data in children's brain tumor network has been de-identified, so it can't be directly associated back to a single participant. And all of it is viewable within the browser within Flywheel. And then you can also run analysis workflows, so you can process files and segment them with specific protocols or workflows within Flywheel as well. And then this table over here on the right shows the number of different sessions that we have based on tumor type. All right, so at this point, I'm going to take a brief break from my slides, and I'm going to show, we're going to dive into PCBio Portal together, and we're going to compare and try to answer 2 of these research questions. And so you can follow along with me. You can answer these questions yourself and many more at our website, pcbioportal.kidsfirstdrc.org. So I'm going to exit out of my slides, and I'm going to navigate over to my other tab, to pcbioportal.kidsfirstdrc.org. So first, I'm going to go to the Pediatric Brain Tumor Atlas, the Open PBTA study. So this is a large collection of curated CBTN data. This is what I was showing in my screenshot previously. We're going to wait for it to load. And so here within Open PBTA, it kind of can be a little intimidating if you dive in cold, or it just throws a lot of tables and bar charts and pie charts at you very, very rapidly all at once. But I'll walk you through how to kind of begin to tease this apart and understand a little bit more about what we're looking at here. So you can kind of think of it like if you go to the library and you're looking for a specific book. In the catalog, you might add filters. Well, I'm looking for a book by this author, and it was published in this year, and it has this title. And so you can imagine that you can add these filters, and then that would narrow you down towards your particular book you're looking for. So it's a similar type of setup here. Well, here we're looking at genomic data of CBTN patients. And so right now we're looking at 943 patients, and associated with those patients, there's 1,122 samples. And so each of these graphs and tables represents data corresponding to all of those particular patients. So we look at, for example, mutated genes. It tells us these are the most frequently mutated genes across our particular selection here. The most frequently, P53, H3F3 alpha, BRAF, and so other very frequently arising oncogenes. Our tumor suppressor genes are showing up here in our list. But you can also search for very rare genes as well, or for example, if you're interested in EGFR, which we couldn't see at the top, we can begin to type it in and find it in our search there. You can also, it provides information on, for example, patient demographics and race and ethnicity, as well as tumor types and tumor histologies. And so, again, to go back to my library example, you want to begin to apply filters to be able to narrow down. And so, for example, I can select, maybe I'm interested in P53, and so I can add a little checkbox there. And then I can apply and select just these samples. So now you see everything is, my wheels are spinning as we are applying this filter to be able to look in more detail at just the samples that have P53. So once this loads, then we'll be able to see an updated view here below based on just looking at those particular samples. It's taking a little bit longer than it normally does. So I might back out of here. go back to our whole view, but I will go back to our question that we were interested in answering is how do mutations in P53 influence outcomes for diffuse midline glioma patients? So if I go down, I can select our diffuse midline glioma patients. So now we're just looking at 103 patients who have the histology of diffuse midline glioma. And so now you can see my numbers have updated here. I just have 103 samples of 55 patients. So we've narrowed our focus. We've applied that filter on the library catalog. And I'm interested in P53. So what I'm going to do is I'm going to query this. So you can either check it in your gene table, or you can just type it in the search box up here. And we're going to hit query. And so basically what that query button does is it says, tell me everything about the genes in this box about this set of patients. Query. And so this is what the computer is currently processing and trying to answer for us. And so what we have here, so this is our list of it's called an oncoprint. So each of these columns corresponds to a specific participant. And then each of these, little dominoes corresponds to, is color-coded according to this key. So it's similar to for example, a genome browser with different tracks. So you could add additional tracks. We could look at the age of the participant. And then we get these bar charts that show, older patients versus younger patients. So you can filter and view kind of an overview of data here. But what we also see is that we see that 69% of participants with diffuse midline, of our diffuse midline glioma samples have a mutation in P53. So our question, to go back to it, is how do those mutations influence patient outcomes? So if this, 70% do have this mutation, 30% don't. So how could we compare, this group of 70% of patients to this 30% over here? We want to compare this versus this. So that's done under the comparisons tab. And so what we can do is we can compare within what's called the altered group, or more specifically, we can call them the P53 group. So these are samples with alterations in the gene P53. And then we want to compare them to this other group, this unaltered group. So this is a set of participants who have diffuse midline glioma, but do not have a P53 mutation. So we want to compare yellow versus blue. And what PCBio Portal does is it makes this really easy. So once you kind of have these groups defined, this is yellow, this is blue, what's the difference? It does automatically generate a lot of comparisons for you. So for example, we can pull up survival. So how is survival influenced by whether or not you have P53 mutation? It is. So here's the Kaplan-Meier plot where we can compare yellow and blue. We can see that patients who do have DMG with P53 have worse outcomes than other DMG patients. And in fact, it already does the statistical test for us. And you can download this figure as a PNG and save it to share it or include in a presentation and export this information as well. We can look at additional clinical data as well in terms of, are there any other things that are correlated with being in the yellow group or in the blue group, as well as genomic alterations in gene expression. And so you can begin to find other comparisons, things that also correlate with being in one of these two different groups or not. So there's our answer to our question. How do mutations, they do influence it, and patients with mutations in P53 have worse outcomes. So we just answered that question here today. So question 2, which we can answer together, is how does expression of GLI1 vary among the medulloblastoma subgroups? And so this is a question that we're going to answer here. We're going to go back to CBio Portal, and we're going to clear out our filters, and we're going to start from the very beginning again. And so our question is specifically looking at the medulloblastoma subgroups. And so we're going to select down here in histology. We're going to excuse me, no, we're going to go to cancer type detail. And we're going to see that within cancer type detail, if I begin typing medulloblastoma, you can see that we have within medulloblastoma, if you're unaware, there's 4 different classifications or subgroups. There's medullo group 4, medullo group 3, medullo SSH, SHH, and medullo Wnt. And so each of these, speaking back to the beginning of the presentation, there are different tumor types, there are different tumor subgroups. So this is within medulloblastoma. These are the 4 main categories of different tumor subgroups. So maybe we are medulloblastoma researchers and we're interested in comparing these 4 different groups to each other. And so would you believe it, there's a very easy button that we can click that says compare these 4 different groups. So it's going to send us back into that comparison view, where now we're looking at, instead of having blue and yellow, our colors have grown a little bit more. Now we have 4 different categories, and we're going to compare these 4 different groups to each other. And so looking at, you can pull up survival. If we narrow down our months here, then we can get a little bit more of a view there. But specifically, our question today is we're looking at a particular gene. We're looking at, does the expression of this gene vary among medullo subgroups? So I'm going to go to mRNA, and we're going to let that load. So it does take a minute to load. There it goes. But remember, we are looking at the expression of every gene in the genome for more than 100 different samples right now. So it takes the computer just a minute to think about all those different numbers. Okay, so we're looking at expression of GLI1. So that's our gene of interest, and it automatically comes up with our list of all the genes in the genome. But we're not going to scroll. We're going to use the search box here, GLI1. And it turns out that, yes, there is a higher expression in one particular subgroup. And so, for example, I can go back over here, click on the gene in the table. I'm going to click on it, and there we go. And so if you are a medulloblastoma researcher, you perhaps already knew the answer to that, that yes, GLI is specific. It's a specific biomarker that's associated with the SHH activated medullo subgroup. And here's our box and whisker plot, which shows that expression. It's highly significant. And so, but hopefully by demonstrating how easy it is, I just typed in the search box and typed in the name of my gene. It's very easy to search and explore. And so without, if you have additional genes that you're interested in or your particular gene of interest, you can very easily search and compare across these different tumor subtypes. So there's my example of looking at different gene expression within the CBTN genomic data. So CBio Portal is very exciting because anyone can log on. And so, we talk a lot at the beginning about making resources available for free. So CBio Portal is open access. So this information can be accessed by anyone on the call today. Can navigate to pcbioportal.kidsfirstdrc.org. It's free to create an account. And then once you have an account, you can access the Open PBTA study and begin doing these same analyses. So there's no application, there's no embargo period, there's no data requirements, forms that need to be signed. Anyone can access this today. We're sharing this with everyone who we possibly can. So I love our CBio portal. So please, after the talk, I hope you'll consider exploring CBio portal a little bit further and using that to continue your own discoveries. All right, so I see that I'm coming up on time here, and so I will close by highlighting some of what we call our collaborative resources. So how do we collaborate and how do we share together, and where are some next steps to continue the conversation? And so our biospecimen and data resource, we provide this through these online portals. If you are interested in accessing our open access data like PC Bioportal, again, it requires no additional contact, additional forms. Just navigate to the website and begin your exploration. We do, of course, we do make the whole genomes available as well as, of course, the biospecimens, which require some conversation and collaboration as well. But all this is 100% free for any researcher who works at any institution. There's no hidden fees associated with it. And we encourage investigators who are looking for more information about a particular set of biospecimens or a set of genome files to reach out to us at research@cbtn.org. So this is the best way to get in touch with us. So in terms of how to apply for access, I would point you to our website, which is another great place to follow up, which is cbtn.org. And so if we pull up a new tab here, we could go to cbtn.org. And so this is our homepage. It provides information about all of the different projects that we support, our different websites, as well as the available resources that we make available to investigators. So cbtn.org is another great place to get started if you're interested in learning more. Additionally, in terms of if you're interested in submitting a data application, so if you're interested in accessing those biospecimens or the cell lines or the controlled access raw genome data, you can submit click the application form, which is right on our homepage, access data, and that brings you to our request. And so this one request form covers biospecimen requests as well as data use requests as well. And so you can click through this red cap form, and once it's submitted, members of our team will follow up with you with either follow-up information or information about how we'll deliver what's requested as part of your application. So definitely check out cbtn.org, our homepage, after our talk today. Back to my slides. So our timeline, and so sample portions of the request will be reviewed by our team. We can also help you find a cohort. And so in terms of if you need help using the sites, we can help identify participants or samples that meet a particular criteria of your own as well. We try to turn those around within 2 to 3 weeks upon receiving them. For further, especially for our biospecimen requests, those are submitted for what we call primary scientific review. So this is our scientific committee who will review them. These are investigators at our institution hospitals, and they will return a decision typically within 4 to 5 weeks. If everything is approved, then we can ship samples within 6 to 8 weeks upon receiving the request. And again, I'll emphasize that beyond just the website, research@cbtn.org is the best place to get in touch with us after the talk today. We're also available on social media as well, so please follow us. We're at CBT Network on all major platforms. We use our social media to help promote CBTN research. So we highlight patient stories, we highlight investigators in our investigator blog posts. We highlight scientific publications using CBTN data, as well as conference presentations as well. Ultimately, we want to share incredible stories and incredible breakthroughs with the world. And so if you use CBTN data, we'd love to continue to promote your work with folks who will listen to us. We also make available monthly office hours series. This is what this is kind of a casual conversation where we help tackle users' questions. If you're interested in learning more or not sure how to get started, all the way to I did an analysis and this is what I discovered and I'm trying to figure out what this means. So we really try to make it as collaborative and open as possible to be able to share. So we hold them once a month. Unfortunately, we're not going to be able to host our December ones, but they will resume again in January, and details will be available on social media. So follow us on those networks or ask research at CBTN, and we can have you added to our recurring calendar invite. Another collaborative resource we offer is our PNOC CBTN working group. So PNOC is the Pediatric Neuro-Oncology Consortium. PNOC is specifically focused on improving brain tumor treatments through better clinical trials, which is our shared goal as well. So we are very collaborative partners with the folks at PNOC. As part of PNOC, they have the CBTN PNOC working groups. So this is a collection of investigators who are, working together, collaborating, discussing, holding monthly meetings to kind of tackle a specific problem or a specific diagnosis. And helping to move us closer to that ultimate goal of better clinical trials and better treatments. So if you're interested in participating or learning more about the different working groups that are available and joining these meetings and joining this community of researchers, again, research@cbtn.org is a great place to email, and we can provide information about those meetings. And then finally, I'll close with our collaborative project, the Open PBTA. So this is a set of investigators who are all sharing our open science interest in sharing and annotating pediatric brain tumors. And so someone will have a great idea. We'll say, great, let's do it together. We'll generate the data, and then we'll make the results of that analysis available. And so if you have a specific analysis in mind, it might already be done. And the folks at Open PBTA would be happy to share that process data with you. If you have an idea and it hasn't been done, then we'd love to work with you together. We'd love to learn more about your method, about your research, and we'd love to run the CBTN data set through it. These folks at Open PBTA were very active in GitHub within Alex Lemonade's stand repo/Open PBTA analysis. This will be a great place to learn more about joining our collaborative open science work. So that's the end of my talk. And so hopefully this was a great overview of what we have available at the Children's Brain Tumor Network in terms of our resources that we're sharing with researchers, investigators, clinicians around the world. We really hope you'll follow up and connect with us. Again, one last time, our website is cbtn.org. You can follow us on all major social media platforms at CBT Network. And then finally, you can email us research@cbtn.org. This is the best place to talk to our operations team in order to learn more about submitting an application or for data or biospecimens. So with that, I'll close it here. I want to provide one more thank you to the folks at St. Baldrick's for helping to organize this talk today. Hopefully it was clear how our CBTN resources can help empower investigators around the world to be able to make great discoveries for kids, which is our mission. So with that, I'll close. Thank you so much, everybody, for attending this talk today. We look forward to working with you, hearing from you, and collaborating with you in the future. Thanks so much. Have a good afternoon.